have here is shared worldwide free of cost. So thank you. You just introduce yourself.
What's up? Oh yeah, I'm Jeffrey Neuropathology. I'm a clinical scientist and neuropathologist, leader of the neuropathology report, as well as the brain program, and have a lab in data science and neuropathology. For just like a show of hands interaction, how many of you kind of know in pretty good detail what a pathologist does? How many of you know a pathologist and work with them kind of routinely? Yeah, so that's exactly why we work these interactions. We study things at cell and tissue level, so everything you're doing on socioeconomic, behavioral, lifelong exposures, that has real biological effect that we're trying to understand and measure from the microscopic perspective. And I always start with this because even though the Alzheimer's center patients, I know this serves as their final position, final interaction with us as a diagnostic provider. But it's still all about the patient, and that's our mission, and that's our goal. So that's my infant daughter, and she was a toddler, and my wife, mother with advanced dementia, and just spanning almost 95 years, there's still that human connection, even though her memory is long since gone. And that's on the right. On the left, my neurobiology professor from the University of Iowa. A recent photo, I kept in touch with him over the years. He was a great mentor, and my wife gave permission for me to share this. And he developed Lewy body dementia, and we're all touched by this in some way, and we're all contributing our expertise to help in this fight. And so many of you are already familiar with this, but the clinical challenges in age-related dementias is that they're heterogeneous and overlapping. The clinical symptoms often reflect the neuroanatomical site of involvement, and many different biological, pathological processes can affect the same set of regions and networks. So that creates challenges with the diagnosis, and they often co-occur, as Toby said. In sporadic age-related disease, co-pathology is generally the rule, not the exception. The vast majority of cases we see at our Alzheimer's Research Center with sporadic late onset dementia has more than one pathology. And the clinical diagnosis, based on that symptomatology, based on historical data and testing, can be discordant with the pathology of the primary diagnosis at the cell molecular level 30 to 40% of times. And what that means is that it's not necessarily wrong per se, but it's incomplete, or it's not a primary Alzheimer's process, it's the primary synuclein Lewy body process of secondary Alzheimer's. And why does that matter? Because when we're developing prognostics and treatments, we want to treat the full spectrum of the biology of the disease. So if they're receiving anti-amyloid therapy, but they have synucleinopathy primary, your clinical trials aren't going to be empowered, and they're not going to benefit from that as they would if it was a primary Alzheimer's process. And for non-Alzheimer's dementia, there are fewer antemortem prognostic blood, CSF, and PET biomarkers. And so in order to get better treatments, we need to identify these patients into their appropriate disease subgroup, their biological disease process, and stratify them to the correct trials, either Alzheimer's only treatment, potentially Alzheimer's with synuclein, TDP, targeted treatment, and mixed, and all of that combined with lifestyle factors and helping vascular and other cerebrovascular risks. And neuropathology remains the gold standard despite all of these fancy blood, CSF, and biomarker challenges. That's again because of limited resolution in limited sensitivity and resolution in these tests. Blood tests, we can have things. There are blood tests for Alzheimer's currently, but that doesn't measure the non-Alzheimer's processes. It's also uncertain how sensitive that is to preclinical or clinical disease and what sort of threshold matter. PET biomarkers are great, but they don't pick up very early, and it's a limited resolution, and we need a relatively high burden of disease pathology to see it. And so we still need a way to study and diagnose at the cell and tissue level. And the key message here is that this serves as kind of the target or the ground truth for everything you all are doing, the understanding the clinical trajectory, understanding the biomarkers. And my goal is to help the neuropathology related classification and understanding so that we can predict future cognitive trajectory, and we need that with these antemortem studies that combine the postmortem ground truth. A principle of the pathophysiology of neurodegenerative diseases from the pathologist and cell biologist perspective. There's kind of three principles that I teach and tell all the Chinese students. There's a stereotypic neuroanatomic region involved, and a characteristic spread. So tau often concentrates and begins in the medial temporal lobe and spreads out. Amyloid starts in the neocortex and then goes into the deep gray nuclei, nuclei and cerebellum. There's selective regional and cellular vulnerability. So not only are certain sites involved, within those sites, certain cell types are preferentially vulnerable to the pathologic processes. And finally, the pathologic aggregates. So kind of these three principles are stereotyped regions of spread, selective vulnerability, and pathologic aggregates are kind of three core themes of all neurodegenerative diseases, from Huntington's to Alzheimer's to any other type of age-related disease, TDP-43 motor neuron disease, etc. And so we can look at this as kind of the organ, tissue, cell level. And starting from the ground up, the molecular level, we know these are abnormal protein conformations, conformers that tend to aggregate. And that does not mean that they're going to be spread through contact and handshakes in a way that, a true prion disease would, but they do propagate sort of within an individual at a very low, slow level. And you can even look at them at the ultrastructural level, and they have distinct structures by electron microscopy, which is kind of unique. And they aggregate in certain cell types. That's the selective cellular vulnerability. Tau hyperphosphorylase aggregates within neurons, within dopaminergic neurons. Alpha-synuclein aggregates form these Lewy body disease, and also within cortical neurons for cortical Lewy body disease. And then also spread in the same way. So kind of general synucleinopathies, neuronal synucleinopathies like Parkinson's, Lewy body disease often kind of begin or start in the brainstem and kind of spread up. So that's why you get Parkinson's disease dementia. It starts in the brainstem and then spreads up to involve the medial temporal lobe and cingulate, and you get the behavioral diseases. Whereas, Lewy body disease may have a more heavy early involvement within the cingulate, within the medial temporal lobe and amygdala region, while still having the brainstem component. And thanks to decades of research in Alzheimer's, we've made tremendous progress towards earlier diagnosis, better understanding. There are now blood tests, FDA approved. There are CSF tests for the amyloid tau ratios. Again, it's focused on that pathologic aggregate that's often specific to certain diseases or shared among the limited subset. And there's PET for both amyloid and tau. For Parkinson's disease, Lewy body disease, there's no blood tests. There are CSF amplification assays, and there's no PET tracers. We still need these PET tracers because we want to see, again, where it's spreading, how much. The biofluid tests are great, but they only give you a static kind of bulk count of what's there. And for diseases with TDP-43, like ALS, like frontotemporal lobar dementia. There are no blood tracers, and there are early research level work into PET tracers, but nothing that would be ready to deploy clinically and have a high enough sensitivity for this kind of rare and low abundance cardiomyopathy. And so, why does brain imaging matter? I think you all, have an idea of this, and the survey is the ground truths. It's largely inaccessible during life, and I think that's a great thing. I don't want anyone to have brain biopsy. There's just no need. And at the cell molecular level, it's really a disease, these abnormal protein conformations, how they affect the vulnerable neuron population, and that defines the ground truth. So we still need to look at cell molecular level in the context of the native tissue and this brain. The native tissue serves the validation for these important biomarkers. The only reason we have amyloid tau PET is because of generous brain donors over the last 2 or more decades who have, given their tissue to science and supported this research to prove, yes, this compound does bind to this protein in real, in a real setting, real tissue. And then we can also do, exciting research in collaboration with all of you and other cell and molecular scientists, looking for spatial patterns and understanding why are certain neurons resilient versus vulnerable. Why is this population affected, but a neighboring one is not? And it also, has data to share with other researchers and do data mining and try and understand this using multiple modalities. And so the way I see it, is it's not just the neuropathology for the sake of itself. Find better these subtypes to give you all clinicians and biomarker scientists a better target to compare your clinical pathologic correlation. And so precision medicine, often begins with the molecular basis of disease, and that's why I feel that that pathology and fluid and tissue-based studies are so important to get a really deep understanding of that individual disease process using multiple modalities, genetic, clinical, all sorts of survey and other metrics, exposomes, and then the tissue level understanding. As I said before, the neuropathology kind of is in the realm of finding that cell and molecular disease process and really accelerate other research. And current neuropathology frameworks are not really doing, I think, you all a service when you request, patient information for people who've donated and had their neuropathology diagnosis. We take, many dozens of brain regions. We do deep, detailed analysis, by trained experts. We do dozens of stains and histologic tests. But at the end of the day, we just give you the ABC score. And so we're taking, multiple data, spatial information, structural and other information, and we're just kind of not using it. And that's just the product of not having the right tools historically to quantify that in detail. And so we're working on precision neuropathology and collaborating with other centers who are doing really advanced processing, integrating with neuroimaging, antemortem and postmortem. And so this is some of the work that Toby mentioned. We're doing sort of precision, not quite stereotactic, but trying to get close to it. Thin slices to get precise anatomic subregions. For these cases, we're also getting ex vivo MRI, so we can correlate that to the imaging scans the patient had during life and have a kind of final point for that structural trajectory. We're doing some machine learning and other studies to try and get ways to reconstruct the cortical surface, to measure atrophy, gyral atrophy in a more granular detail. And as Toby was showing some Toby's work, the precision neuropathology enabled some more PET validation, enabled these biomarker studies. So an antemortem tau MK6240 from the patient, and then from the same patient, the donated they took a similar region of cortex and did autoradiography with the tau compound and tau immunofluorescence. And we're seeing here, kind of at a very high resolution, what is this tracer binding to, what specific abnormal forms of phosphorylated tau, and how does it correlate with measures such as the SUVR for the PET and the abnormal tau 231, 396, these different tau phosphoepitopes. So understanding this tracer at a deeper, much deeper level, and you can also use it to find new tracers. And that's what we're working with Henrik Zederberg, Dr. Kuhn, and Art Cole, using the tissue, doing precision dissection to give specific diseases and subregions for proteomic discovery to find signatures that hopefully will be able to translate to antemortem fluid and other biomarkers, so that we can find signatures in the disease state brain, correlate them with the blood and fluid tests we get from patients during life, and link them to find a shared signature of a peptide or expression, such that we can now use the blood to predict the end state, or at least give an estimate of what the end state might be. And then we're with Artemis Cole, a leader in like chemistry and developing new PET compounds, led the development for here. We're now trying to find PET ligands for nuclein and for TDP43. So we can measure the disease spatially, monitor it over time, the spatial progression and abundance. And again, we're using the donated tissue to find, as close to a intact tissue setting, what will bind and what won't. And this is the slide of our Toby, focused on the antemortem biomarkers that are now incorporating these biological staging of amyloid and tau in neuronal injury. And we can characterize them along the spectrum of disease burden and clinical symptoms, and identify patients who are maybe progressing faster than you might expect given the burden of amyloid and tau, or resilient and have a high burden of amyloid and tau that to others would cause a severe clinical dementia, but to them is either sort of unimpaired or mild cognitive impairment. And future studies using these data generated from the antemortem postmortem correlation, these biomarkers, we can now try and study, what makes people resilient. How can we improve resilience, cognitive resilience, cellular resilience, generally in the population? And to do that, we need precision markers for the other protein aggregates that we don't have. And then the final thing I'll say while on this slide is that there's still, once we have all of the biomarkers and other things that we could ever wish, I think there's still a role for neuropathology, at least currently, because once we give these treatments to patients, we need to see what it's doing. Is it actually removing amyloid and tau? Is it causing inflammation that could be harmful in certain ways? And so, as we transition from having the biomarkers and having more precise clinical and more diagnosis, there's still a role for generous donors to help teach us what these treatments are doing at the cellular molecular level, making sure they're causing more help than harm. And with that, I'll thank all the collaborators, all the brain donors, everyone who followed me, the Alzheimer's Center, and our program, and lab, and external collaborators.
Any questions?
Done. Everyone here proceed.
Thank you, Jack.